Hello, my name is Sarah Hearne, and I'm here to speak to you today about using DSI to interrogate crop genetic diversity for climate change adaptation. Specifically, I'm going to speak to you about the Allele mining initiative. This is an initiative that is operated by a multitude of CG centers and US universities, focusing specifically on 6 species: maize, sorghum, rice, cowpea, cassava, and common bean. This endeavour is currently funded by the Bill and Melinda Gates Foundation and FAR, and it maps as a sprint to the Agricultural Innovation Mission for Climate, or AIM4C. When we think of the germplasm banks, we inherently think of all of the diversity contained within them. And this diversity is a treasure trove for modern breeding. All of the native variation that we could possibly sample is predominantly held and maintained in these banks for humanity. But when we look at these collections from the breeding perspective, as you see here, some of the maize collection held at CIMMYT, while we see an array of colour, seed shape, seed size, etc., it is very difficult for us to identify which of these collections may hold diversity that is novel to our breeding pool for climate change adaptation, for example, for heat tolerance or for drought tolerance. All of these collections contain passport data. Now that passport data, just like your passport, my passport, details some characteristics which may be useful to help differentiate these collections. In this case, we look at the maize collection of CIMMYT, and each of the dots present on this graphic indicate the number of accessions that have been collected from a site within the hexagons that you can see coloured on the graph. Now, this location data can be very useful. Just like a place of birth, the place of collection of the accessions can be used to derive other features. In our case, we've used these collection sites where they are precise to pull out long-term climate variables of potential interest for breeding. So for example, here you see average annual temperature graphics. We can use the collection site origins to identify the environment under which the particular accessions have evolved and have been selected by farmers. And using the paradigm concept that if something has been evolved and has been selected under a particular environmental feature, it should have adaptation to that feature. So for example, accessions identified that have come from environments where there have been a predominantly high temperature or a high temperature regime during the crop growth cycle, those accessions should have greater tolerance to that particular feature. Working under a previous initiative, the Mass Agro or Seeds of Discovery initiative, many of the collections have been genotyped. So we have genomic data, genetic fingerprint data that underlies these collections in addition to climate features. Now what we are able now to do is extend proof of concept work that has been conducted in maize and sorghum and conduct what we call environmental GWAS. GWAS is simply genome-wide association studies. And what GWAS studies typically do is take a phenotype of interest. In humans, that may be high cholesterol level, and you look for associations of that with the genomes of different individuals that have both a genotype and a phenotype. Now, in this case, we do not have a direct phenotype, but what we do is use that climate data, the collection site climate data, as a proxy for adaptation to particular environmental regimes. Now we've run these analyses in maize, looking across 17,000 accessions for heat and drought. And what we see are features like this. So in the upper graphic, what you see here is part of a genome scan. Moving across the x-axis is like walking along the chromosome of Maize. In this case, it's chromosome 1. And as we move from left to right, you're moving further and further along the chromosome in position. Each of the dots here represents a molecular marker or a molecular signpost, part of the genome where we are able to measure different levels of variation. On the y-axis, you see the association of each of those positions, each of those molecular markers, with a feature of interest. In this case, it is precipitation in the growing season. The further up on the y-axis that peak is, the more significantly associated it is with the feature of interest. So those 3 dots above the blue line, they indicate that there is a significant association in that region of the genome with precipitation under the growing season. When we look at this region of the genome and we ask the question, what genes are involved? We start to find genes. In this case, you can see one on the lower part of this graphic. This gene is involved potentially in adaptation to drought stress. It's involved in plant cell signaling. Now we can then take that gene and we can look at the 3 different forms that we are able to measure using our data. So you have the form that is little a, big A, little a, and big A, big A. And here we have taken data from a trial from about 1,000 different entries, and we have grouped those entries solely on the form of their genotype at that particular part in the genome. And as you can see, looking at the y-axis for drought yield here, all of the particular entries that have a big A in their genome, whether it's big A or big A, little a, they have higher yield under drought stress than the little a types. Now, in many cases, breeding has done an excellent job of already identifying and harvesting these kinds of genetic variants. However, many have not made it through the breeding bottleneck. And from initial analyses, we see that 7 to 8% of the variation that we find running this kind of analysis, variation that has potential adaptation to climate change, such as drought and heat stress, these variants are unique in maize landraces. They have not made it through the breeding bottleneck into modern varieties. Therefore, there is genetic variation that is of value for breeding that remains on the shelves in the many vaults around the world. Now, identifying this novel variation is very promising. However, what we then do is then repackage that variation into a background that is more accessible for breeders across the globe to utilize in new varietal development. So what do we start with? Well, we go back to the landraces, and we see that this novel variation is not found in 1 or 2 landraces. It's found in a multitude of accessions from different regions and countries. And it's only through this cross-boundary analysis of thousands and tens of thousands of accessions that we can identify these variants. So the power comes from having integrated access to data across global collections. Now, moving forward, there's another very interesting thing that we can do with this information. We can take the same base data, that's climate data, and the genotypes from these geolocated accessions in the banks, and we form a training panel. And what we can do with that training panel is extrapolate to identify a novel climate that we have. So in this case, there's a particular area on the Yucatan Peninsula that I may be interested in breeding for. So we take the climate features of that site, either today's climate features or using climate modelling, we take features of that site that we think will be predominant in 20 years, 30 years, 40 years in the future. And we feed that climate information into the training model. And then what we pull out from that model is the genotype that is predicted to have the best environmental adaptation to that site. And when we mean adaptation, we're talking about the ability to reproduce. So we're looking at reproductive biology, and in the case of seed-based crops, we're looking at some level of proxy for yield or performance. The model generates a series of predicted genotypes. These genotypes may not exist in reality. We don't have those recombinations of genes available in the gene bank, but we may have things that are near to it. When we validate this particular method, here we have a little graphic that looks at the validation within existing trial data. So this is real data from trials run at this location in the Yucatan Peninsula. On the x-axis, you see the distance of the entries that are present in that trial, their genetic distance from the predicted genotype, the genotype that is predicted to have the best performance. So as you move from left to right on this axis, you move from materials that are more similar to the predicted genotype to those that are really very, very different or very distantly related. On the y-axis, you see grain weight in tons per hectare. So this is a value of yield. And as you can see, that blue line and the grey boundary around it indicates an incredibly good correlation between the genetic distance from the predicted genotype and the performance in the trial. So this indicates that the genetic diversity of germplasm collections, when explored at scale with modern tools and with a diversity of partnerships, offers a very powerful tool in our efforts in breeding to mitigate the impacts of climate change. Now, throughout this work, we have a proactive policy when it comes to data stewardship. We are working to ensure that the data remains in the public domain as an international public good, to ensure that the data is as accessible as possible to as broad a range of stakeholders as possible, whether they're using tools themselves or they need tools to aid interpretation and use of the information. At CIMMYT, we operate using data licensing for this kind of DSI information. And we use CIMMYT instances of tools called Dataverse and Germinate, two platforms that enable us to release that information publicly with associated licensing. This licensing allows the use of the data for research, educational, and scientific publishing purposes. We try to endeavour that the user shall not seek any form of intellectual property rights or other legal rights over the data accessed, ensuring that that data remains free for everybody to use. And we request that the data may not be distributed in any form without written consent of the provider, ensuring that the data is accessible from one source with the associated licenses. The data is available and the knowledge that's generated from this data is available in processed and raw forms, reflecting the different abilities of stakeholders to access and use raw data. Sometimes we pre-process, so it's easier for those who may not have the tools and computational resources to make those translations. So I hope here we've demonstrated some of the immense value of using DSI across these particular collections, and have tried to demonstrate that within the space of DSI and data stewardship, we're doing our best to make data available while respecting and reflecting potentially absence of some framework within the multilateral system for data sharing. Thank you very much.